Good morning, everyone. My name is Miss Topion, or you can refer to me as Miss T, whichever makes you feel comfortable. I'm fine with both. I'm going to be your ninth grade biology teacher, and today I thought I'd give a cute little demo lesson, cool demo lesson on genetics, actually Mendelian genetics, and we're going to delve into that in just a bit. All right, let's get going. Okay, so I like to put today's objective on the board right over here, but because I don't have a board right now, I'm just going to state it right here, but typically it will always be on the board. So today's objective is going to be create a punnet square and predict the phenotype and genotype of the given cross on a piece of paper that I'm going to hand out. Or you can write it on your own paper, doesn't matter. And the second thing I'd like you to do is to define at least 3 of the following 5 terms: trait, allele, punnet square, genotype, and phenotype. We're going to get into what all of this means, so that's why I like to keep it up on the board so it can be clear and you can know what's expected of you. All right? Okay. So I like to start my lessons with a do now, and we're always going to have a do now. It's going to take about 5, max 10 minutes every day at the beginning of class. So for this do now, I want you to take 5 minutes to read this article that I handed out to everyone on selective breeding for pumpkins. And it's totally okay to discuss it with a classmate as long as you're quiet and whispering. So while you're reading this article, I want you to keep some questions in mind, like what do you think about it? What do you think is cool, intriguing, interesting? Did you know about this already? Is this related to something that you know about? Just some things to keep in mind. So go ahead and read it. It's a quick read. All right. Now that you guys have read it, is there anything interesting that you guys thought about the article, or can anybody give me a quick summary of what happened? David? These people, yes, the people bred pumpkins to make them bigger and bigger and bigger. So they had these competitions, 10 feet long pumpkins, exactly. Is there anything you thought that was interesting about that? You're wondering how they did that and so on. Yeah, good question. We're going to get into that today. Anybody else? All right, cool. I think is awesome about it is that they take the biggest pumpkins and keep mating them together and for certain characteristics and traits to make them bigger and bigger and bigger. Just like back then, we used to do this all the time because we didn't have that technology. So I'm going to delve into that right away on how we used to do it back then. So throughout history, we always wanted to know how traits were inherited from one generation to the next. Many offspring look like one parent or the other. You might often see that. But generally, we all look like a mixture of our parents, or, plants kind of look like a mix of whatever they came from. So it's a blending. So people back then engaged in like controlled mating. So they would mate fast horses with fast horses to hope that they get those desired traits, a faster horse or big oxen with strong oxen to make even stronger. And they did this with pumpkins and fruits and stuff too. I don't know if you know, but back then our corn used to be this big. Now it's like this big because through mutations, we would just keep picking the bigger ones and the bigger ones and mating them and making them bigger and bigger and bigger over time. But they didn't quite get why this was happening or how this was happening. So it was just called selective breeding and hoping. So what the real question was what will be the outcome when you mate a particular individual with someone else? Like if I took this guy and this girl, what exactly are their kids going to look like? Or if I took this fruit and this fruit, what exactly will the offspring look like? And all of this ties into something called genetics. Now, can anybody tell me what the definition of genetics is? Harry? Chromosomes? Close, it does have to do with it, but not quite. Genetics is the study of heredity and the variance of inheritance. And can anybody tell me what heredity is? Anyone? Well, heredity is how traits are passed from parent to offspring. As you can see, it took a bit of time, I'm not sure why, but the definitions are both up there. And you can see a cool little comic that says, "Why the long face?" And he goes, "Heredity, it's passed down to him." And you see in the middle, that's something called the Punnett square that we'll talk about. To the left, is going to be, it's a DNA strand, which is pretty cool looking. And the heredity should pop up any second now. All right. Heredity is, oh, I had to click it. There you go. Sorry about that. So heredity is the passing of traits from parent to offspring. All right, we're going to learn way more about this stuff. So when we think about genetics and heredity, there's always one scientist that should come to mind, and that guy is above me, called Gregor Mendel, or we just refer to him as Mendel usually. So he's known as the father of genetics, and he basically is the founder of modern day science of genetics. So he's most famous for his work with pea plants all the way back in 1865. And unfortunately, they didn't really accredit him for the stuff that he found out till generations later, which happens often with some scientists. So Mendel came up with 3 laws of inheritance or heredity, and they are called the law of segregation, the law of dominance, and the law of independent assortment. So for today, for this lesson, we're just going to focus on the first 2, segregation and dominance, because law of independent assortment is a little bit more complex, so we'll learn that at a different time. Okay, so for the law of segregation, it is a gene pair defines each inherited trait. So a trait is defined by a gene pair, meaning parent genes are randomly separated to sex cells that contain only one gene in the pair, and the offspring inherit one allele from each parent. So we as people have 2 alleles in us because we're one being, and then if I were to have a kid, I would pass one allele at random, and the man that I would be with would pass one allele as well, and when we have an offspring, that offspring would have 2 alleles at random from us. That's basically what the law of segregation means, and it's at random. Remember that. You can jot down these notes to refresh your memory and just, keep understand what I'm talking about, because it'll help later on when I'm going through with the slides. So we got to find out what a trait means at the very least. A trait is a characteristic or a feature passed from one generation to another expressed by a pair of genes. So it's basically our physical attributes. It's our physical characteristics, basically our hair color, our eye color, hand size, our height. These are all traits of us, and they're expressed by genes. Don't forget the word gene. And these are some of the traits that Mendel actually was experimenting with. So he had tall plants versus short plants, so it was height that he was looking at, but 2 different types. And then purple flowers versus white flowers, so colors. So these were all the different types of things that he was looking at. Let's do a little recap to see that you're following along. All right, so number 1, the scientist known as the father of genetics and the founder of modern day genetics is, anybody? Albert Einstein? Not quite. Not exactly. Anybody else? Yes, Mendel, exactly. Mendel is known as the father of genetics. Number 2, blank is the passing of traits from parent to offspring. Anyone? Exactly. Heredity is the passing of traits from parent to offspring. 3, a characteristic or feature expressed by a pair of genes is called, anybody? A trait. Those are our physical attributes. Those are characteristics. So trait and characteristic are kind of synonymous. So you can try to remember it that way. All right. So now we got to get into what a gene is. A gene is the most basic unit of heredity. If you ever heard of, a cell being the most basic unit of life, well, a gene is the most basic unit of heredity, carries all our DNA and stuff like that. It determines your traits. It's basically what controls and expresses it inside your DNA. So it's actually a section of a chromosome in your DNA where the base pairs code for that characteristic. It's this little section in the DNA strand. That would be your gene. We have 2 copies, which are alleles, you'll see later on, one from a mother, one from the father. So what did Mendel figure out in his experiments? He decided to work on pea plants because they're true breeding, meaning purebred. That means they share the same phenotype we'll learn about later. So it basically means that these plants look like themselves. they breed themselves, they self-pollinate. So a yellow pea plant would make yellow plants, green ones would make green plants. They share two genes for the same trait, so they're always going to look the same. So he focused on traits that he could easily distinguish, like I showed before, wrinkled versus smooth, and stuff like that. And he found that each trait had two alternates. Just like I said, tall or short. He found that there was a difference in each trait. He then concluded that each alternate form of a trait was caused by an alternate form of a gene. It had to be the case. And an alternate form of a gene we call an allele. That's exactly what we mentioned earlier. So alternate forms of a gene are allele. Let's get right into what an allele is. So, alleles are versions of the same gene with slight variations in their sequence of DNA bases. For example, the gene responsible for hair color has many alleles like brown hair, red hair, black hair, blue hair. Not really blue hair, just kidding. But different colors of hair would be the alleles and hair color would be the gene. So we have two alleles for every gene, one allele from our father and one from our mother, just from the law of segregation, how we pass one along, so the kid has two. Therefore, the combination of alleles for a gene turns into a trait. So if you don't get it just yet, I like to use this quick analogy that pretty much makes sense all the time. So, who likes ice cream in here? You? Anyone raise their hand? Me too. I love ice cream. Coffee ice cream is my favorite. Anyway, so if a gene is the ice cream, then all the different flavors like chocolate, vanilla, strawberry would be the alleles. So just because there are all these different flavors of ice cream, does that mean they're not ice cream? No, they are. They're just different kinds of ice cream. So that's basically what alleles are. A gene is still a gene, but there are different kinds, different forms of genes, which would be the allele. So to get more into it, just to be specific, I highlighted with the same color over here what a gene and allele are based upon each other. So a gene is the section in that chromosome where the sequence of base pairs code for the trait in that DNA, and the allele is the actual sequence, not just the sequence, but the inside the sequence, the A, C, U, G type of sequence, or it's T, C, A. All these sequences are all different alleles, but it's still a gene. The gene does not lose its identity. All right. Now let's do a little recap again. Just make sure you guys are following along, no falling asleep. All right, so number one, a blank is the section of DNA that is responsible for the expression of a trait. The section of DNA that's responsible to show the trait. Anybody? Drake? A gene, exactly, good. It's that section in that DNA that makes the trait show. Two, a or an blank is a variant or alternate form of a gene. Just a different form of a gene. Still a gene though. Anybody? Exactly, just like I said, an allele. Perfect. Good job, guys. Okay, back to Mendel. So, since pea plants are true breeding, he understood purebred plants must have two copies of the same gene for each trait because yellow produced yellow, green produced yellow, and a green produced green. So it just kept going on like that. They self-pollinate. But the thing is, he remembers that offspring should look like a blend of their parents, right? I mean, everybody pretty much does. But when he crossed the green plants with the yellow plants, all he got were all green plants. As you can see in the picture over here, they all turned out to be green, even though he mixed it with the yellow. So this made him realize his first law of dominance, meaning some alleles were dominant while some were recessive. And I'll get into that shortly. Okay, so dominant and recessive. I put this little chart above me because I feel like everybody can relate to this. Anybody in here have dimples? See two hands raised? Awesome. That is a dominant gene, meaning if both your parents have it, or if even one of your parents have it, you're going to have it. Anybody with brown hair? See a lot of you. Me too. It's dominant. It will take over if you have blonde hair. Anybody with blonde hair? You have a recessive trait. And we're going to talk about exactly what that means. So, dominant versus recessive. Some alleles are dominant, some are recessive, where the dominant allele is dominant, strong. It masks over the recessive allele. It covers it. So it's like the recessive allele isn't even there, but it is genetically, but it covers it up so you can't see it, like when you look at a person. Now you may be wondering, why is that the case? Why is something dominant? Why is something recessive? Well, it's a little more it's very complicated actually, but you would learn about that in possibly college genetics. But to make it a little simple is sometimes one allele makes a broken protein. When this happens, the working protein is usually dominant. So that would be the reason as to why. Okay, so if an allele is dominant, you must indicate so with a capital letter, like capital B or capital A. Any kind of capital letter would be the allele in a situation when we make Punnett squares. Keep that in mind. If an allele is recessive, you must indicate so with a lowercase letter. For example, the green plants, we would use a capital G. If it's yellow, we use a lowercase g because it's recessive, and we know that from before. And the letter you pick can be arbitrary, totally random, but it has to be one capital, one lowercase, just like I wrote over here, big P, little p, big B, little b, big A, little a. It can't be big D, little c. You can't combine letters like that, but you can choose any letter you want for eye color or something. Just usually when you have brown eye color, you like to stick with the letter B, but you could do D and D if you'd like. Just might be a little confusing. Alrighty, so another recap, just one question. Everybody still awake? All right, awesome. So a blank allele is one that masks and covers a blank allele. Does anybody have the answer here? Derek? A recessive allele? No, not quite. Actually, the opposite. So a dominant allele, it's dominant, it dominates over something else. It covers the recessive allele. And that is what happens, but good try. You guys are doing awesome. Okay, so as for dominant alleles, for a dominant trait to show, you only need one big B allele for that dominant trait to show. For instance, if a father has brown eyes, he can have, we always have two alleles. Remember, every person has two alleles, so we gotta have two letters indicating that. So a father with brown eyes has either BB, big B, or big B, little b. You only need one big B inside there for the brown to show. So both of those mean you can have brown eyes. And the only difference between big B, and big B, little b would mean big B is homozygous for brown eyes, and big B, little b would be heterozygous for brown eyes. I like to remember this by homo meaning same and hetero meaning different. So big B, same, homo. And as for recessive, pretty similar, but a little bit different. So recessive alleles are only expressed if the organism carries two identical copies of the recessive allele, meaning they have to be homozygous for that recessive allele to show. So the two genes we get from our parents have to be recessive. If a mother has blue eyes, which is a recessive trait, her alleles have to be little b, that she has to get from her parents. If it's big B, little b, remember the big B would make it dominant and turn it into brown eyes. They both have to be recessive little b's. And just like here, there's a little example in Mendel's pea plants. So the top one says homozygous dominant because it is the same, two big G's. So we say homozygous dominant, and it's a capital letter, so it's dominant. This one over here is little g, which still the letters are the same, but they're recessive because they're lowercase. And over here is a mix where it's big G, little g, so it's different, heterozygous. However, it still has that one big G there, so it's still going to be green because green is dominant here. Hope you guys are following along. So there's another recap here. Number 1. Can anybody read this out to me? Good. So allele B for brown eyes is a blank allele. Anybody have the answer? Is a dominant allele. Good. Number 2. Allele little b for blue eyes is a recessive allele. Awesome. Now, last one might be a little tricky. Big G is an example of a blank zygous dominant allele. Big G, are they the same or different? Exactly. They're homozygous dominant. Good job, guys. You guys are doing awesome. Okay, so a couple other terms we have to learn about would be genotype and phenotype. So, genotype is the entire set of genes, including the specific alleles found in our DNA. But when we talk about genotype, we're talking about the letters like big B, little b, little b, little b. Here in this case, it's big T, or so on. These are just examples. As for phenotype, it's not so much the letters, but exactly what you see. Like we could say big G, little g is this, but at the end of the day, it's green. It's what we see with our eyes. So it's the observable characteristics, the physical manifestation of all the allele combinations that make it look like that. So an example would be tall or short. And you'll see that soon. We'll be using it. And my favorite part, basically we're getting to the objective of today. We're going to be making punnet squares. Now you may be wondering what a punnet square is. A punnet square is a diagram, square diagram, the line this way, in this way, vertical, horizontal. A punnet square is a diagram that is used to predict the phenotype and genotype of a particular cross or breeding experiment. So on this side, like I was showing with brown and blue eyes, you see on the left-hand side of the punnet square, it's big B, which is going to be brown eyes because they're capital letters and there's at least one of them. And at the top, it's lowercase b and lowercase b, both the same. You need two of them for the blue to show because it's recessive. So they're crossing those two, and inside that punnet square, they're each 25%, and they're seeing what the chances are of the kids having either brown or blue eyes. And right above me are one of Mendel's experiments. So we're going to do a few of these together. Okay, so this is punnet square example number 1. I'm going to do one by myself, then we're going to do like a guided practice working together, and then you're going to be able to do it on your own. All right, so in this particular one, Mendel crossed a true breeding green plant with a true breeding yellow plant, meaning two genes that are the same. So let me just take my marker so I can mark this easily. As you can see here, this GG would be homozygous recessive, and this, we'll put blue, would be homozygous dominant. So what we're going to do is you have to take the column going down and across and fill it in that circle. So I'm going to use, let's say purple. So we're going to go across, write that big G there, and then down. Write that little g there. We're going to go across, write that big G there. We're going to go down, write that little g there. Same thing, across, big G, down, little g. Right over here, same thing. Big G, again, down, little g. And look where that leaves us. We crossed both those plants that were true breeding yellow and green, and we got 4 big G, little g, which is right over here. So now it is heterozygous instead of these homozygous over here, but it's still green. So as I said before, he crossed the yellow with the green and still found green, but they're heterozygous now. So this is exactly what I just did in a better way, but I just wanted to do it in front of you and show you how, but this is clear. Now we have to figure out the genotype and the phenotype. So for the genotype over here, remember what I said, they just want the letters. So they want to find out how many GGs there are here. There's 1, 2, 3, 4, that a little backwards. So we put the number 4 right here. There's 4 big G, little g. There's no little g, or big G, so we don't even put that. Now the phenotype, they want to ask, what do you see? What do those plants look like? What color? So we see over here, big G, little g, we have 4 of them in the offspring, all of these. So it's 100% green plant. So that's what the phenotype is going to look like. All the offspring are going to look green, but the genotype is going to be 4 big G, 1 little g, which is heterozygous. Okay, this is another one where I already solved it, so I'm just going to quickly go over it with you. Same thing over here. Let's choose a different color. So big G goes here, little g goes here, big G goes here, little g goes here, little g goes here, and little g goes here, little g goes here, and little g goes here. So this is a little different. Now you see that we crossed a heterozygous green plant with a homozygous recessive plant. Now let's see the outcome. So now we have, how many big G, little g's do we have? I see 1, I see 2. So we write 2 right here. How many little g's do we have? I see 1, I see 2. So we say we have 2 here. Do we have any big G? No, we don't. So basically we have 2 and 2 over here. So that would be a 2 to 2 ratio, which simplifies to a 1 to 1 ratio, if you'd like to put it in ratio form. As for the phenotypic ratio, they're asking, what does it look like? So now it says, since there's 2 little g's, remember that was the yellow plant, they're recessive. So 50%, meaning this half, is going to look yellow right here, 50% yellow. And these up here are also 50%, 2 out of 4. And that's going to be green. Why is it going to be green? Because it has this one capital letter here that makes it green. So 2 of these, 2 of these, and the phenotype is 50% green and 50% yellow. All right, so now we're gonna do a guided practice together. All right, this time, instead of Mendel's first cross, we're gonna cross 2 of the hybrids together and see what happens. So, Jimmy, can you tell me what the first thing I should do is? Take the big G and put the big G. What should I put in the first box? Good, big G. Awesome. Okay, what should I put in the next box to the right? Samantha? Little g? No. Remember, we have to take this one going across, which is, good, big G, and then take the one going down, which is little g. Perfect. And Jessica, for this one over here, exactly, big G, little g. And for the last one, Franklin, good, it's going to be little g, because you're going across and down. So now we have big G, little g, big G, little g, and little g. Now remember these, they're over here. Now we've got to figure out the genotype and the phenotype. How many big G's are there? Anyone? 1, exactly, right here. So we're going to say 1, big G. Okay. How many big G, little g's are there? Anybody? Out of the 4? 2, good. Right over here and here. So we're going to say 2 big G, little g. Excuse my poor handwriting, I'm doing this with my mouse. And how many little g's do we see? Just 1, right? Right here. So we put 1, little g. So it's a 1 to 2 to 1 ratio, which I will indicate here. 1 to 2 to 1 ratio. And as for phenotype, what do we see? What color are these plants? So big G was green. So we're going to put only 1 is that, so 25% green. Actually, wait, not yet. And then how many big G, little g do we have? We have 2, which means that's also green because it has 1 G. So 1, 2, 3 out of 4 appear green when you look at them. So that means 75% are appearing green. I'll just write a G here for green. And now there's 1 little g. So 1 of 4 is a little g, which is yellow, homozygous recessive. So 25% is yellow. Everybody get that? Yes? Raise your hand if you don't. No? Awesome. All right. We did that, guys. Okay, so now that I did it myself, we did it together. I think you guys have a great understanding of this. Now I want you guys to do some independent work of the objective of what I told you to do. So I want you to create 2 punnet squares crossing any trait of your like, eye color, flower color, whatever you want, any letters that you want. And I want you to figure out the genotype and the phenotype of the offspring, you know, like what it looks like and just the letters, how many. I want you to hand that in at the end of class. And the second part right over here is, let me point right here, is I want you to define in your own words, to the best of your abilities, at least three of the 5 terms over here. So trait, allele, Punnett square, genotype, phenotype. If you could define all 5, that is awesome. I would love that. If you can't, no worries. You could even write how it's a little difficult to understand. Actually, please write if there are any difficulties that you understand in anything. I would love to clarify that for you. If you can't hand in the second part of the definitions because you're busy with the Punnett squares, you can do it for homework and hand it in tomorrow. Absolutely no problem. But I would like for you to hand in the Punnett squares as an exit ticket for when you leave class. Okay, so this is just an example to help you with your independent work. I chose capital round. Anything could be dominant too. You just got to indicate it with the capital letter. So a round pea pod would be big R, dominant, and then the wrinkled pea pod would be little r. So you can cross big R times big R, little r, like this. Big R, and doesn't matter which side you put it on as long as you put them together. And then just cross them the way we usually do, which I will show right here. Oh, actually don't have the one where it's filled in, but exactly, you can do what I said before, any choice of your kind. So key points to take away from everything that we did today, just to close everything up and reiterate what I'm saying. Now, Gregor Mendel studied inheritance of traits in pea plants. He proposed that genes were what expressed traits. Second would be genes come in different versions or alleles. Remember, different flavors of ice creams, but it's still a gene. Genes code for traits. A dominant allele hides or covers, masks a recessive allele and determines the organism's appearance. So when you have a dominant allele, it's going to show, where the recessive allele is going to kind of be hidden if it's heterozygous, or it only shows if you are homozygous recessive. You need 2 little letters from both parents, 1 from each. And when an organism makes gametes, each gamete receives just 1 gene copy that is randomly selected from each parent. Gametes are sex cells like sperm and egg. So we give 1 to our egg, they give 1 to the sperm, they combine, and they have 2 now, just like we do. Lastly, a Punnett square can be used to predict genotypes, all the different allele combinations, and phenotypes, the observable traits of offspring from genetic crosses, what we could possibly look like. So that is the gist of everything. I'm going to give you some time until the end of class to work on your stuff. I'd like you to work independently as much as you can, but feel free to come up and ask me a question if you really need to, if you're really having some difficulty. If you have any help if you need help with homework, feel free to email me, if anything like that. I want you to really think about what you learned today and come to class tomorrow with some ideas of how you like the class or what was difficult to understand, stuff like that. I'm going to put the slideshow up so you can study from it, so you can look at it again and learn. Yeah, so basically I hope you enjoyed my lesson. I had a great time teaching you guys. I wish you guys the best, and